Hello, in this video we will discuss about the pre-organ transplantation test. Yes, so the pre-organ transplantation test is, let's begin to understand with the whole story. So here is the basically the pre-organ transplantation test through blood test, which that will be done in the laboratory. So here is the patient. Patient name is the Mr. Jack, for example. So he is saying that I want to receive the kidney because kidney failure patient. So this is known as the recipient patient. While on the other hand, the healthy person, which that is commitment for donation, so it is known as donor, Mr. John, and he is saying that I will donate kidney for friends. So this is the healthy person, Mr. John. So this is the doctor, which that will check their blood and the blood test for understanding for the donation to the recipient as a patient. So how it's possible? So it will take a blood sample. So when we will take a blood sample from both patient as well as the donor, so it will blood sample containing process we will discuss. So this is the blood sample. This blood sample is basically through injection receive the blood sample from the donor as well as the recipient. So the recipient basically receive the organ, so that is why the blood sample test is very important. So after the blood test will take and go into the laboratory through a lab assistant. So the assistant will collect the sample and go into the laboratory for analysis. So here is a centrifugation of the blood sample from the donor will be create the through centrifugation will separate the serum will light as compared to the blood, so the blood will precipitate down, while the serum will inoculate from this sample, and the serum separation contain immunoglobulin. So remember, this immunoglobulin is specific. So in this way, here is the this is the sample of the donor. So we are analyzing the donor sample contain an immunoglobulin. So when we will take the recipient RBC, one drop, and it will through dropper will put into one drop of RBC to the recipient, from the recipient to the donor serum, which that's contain immunoglobulin. So what will be happen? That's all story will show. What is this? So this is the medium serum, which that's contain a immunoglobulin and incubation 37 degrees Celsius in 1 hour. And this immunoglobulin, if bind with the RBC, so when we will bind with the RBC, so it means it is against the antigen, against this antigen, so the donor antigen, for example. So after the 3 washes, the medium, the centrifugation will process, and in this way, the anti-human globulin will drop, 2 drop will put into the sample, which that is the 1 drop of RBC of recipient and the immunoglobulin containing serum of the donor. So it will be agglutination formation through adds drop of AHG. So this agglutination mean the clotting will be occur. So the clotting like or agglutination formation due to the anti-human globulin will bind with the immunoglobulin, which that is present already. So they're not compatible. So that's it will reject. So if The after 3 washes and adding the anti-human globulin, which that if the no agglutination formation will occur, so the compatible, so the doctor will select, and selection to treat via a surgery through the doctors. So in this way it can be transplantable. While on the case of the not compatible, if we will mistakenly put into the recipient from the donor organ, for example kidney, so the immune system activation, inflammation will lead to adaptive immune response. So how it's work, let's begin to understand. This is a person which that's contain a transplanted organ, and that is against the recipient. So that is why the recipient will produce the antibody against this antigen. For example, this is the kidney, which that is the antigen. Antigen recognized through antigen presenting cell will endocytose through toll-like receptor, and after this entry the antigen presenting cell will go into the lymphatic system, the lymph node, and the naive T cell to show the antigen through MHC class 1 molecule to proliferate the naive T cell into the CD8 and the T helper CD4 cell. So the CD8 is the cytotoxic, will kill the antigen, the kidney, while the CD4 is used for the helping to recognize the antigen for activation the naive B cell after the taking the antigen from the antigen presenting cell. So this naive B cell will proliferate into the plasma cell and B memory cell. So the plasma cell produce a lot of antibody which that will against the antigen as a kidney. So this will collapse, and that is the basically the mechanism for the rejection of the transplantation. So the test is very important. So this is the lymphatic system. So the test is very important for the analyzing for recipient patient to get the organ. So transplantation will be done if the test will be no agglutination, and that will be compatible. While on the other hand the agglutination formation will be occur, it means the organ transplantation is very dangerous, even lethal, can cause death. So thanks for watching. Please make sure to subscribe, like and share. Bye.